So I just like, maybe just make it so I need to.
Okay, everybody, welcome. I see some known faces from my NP1 and just one or two new faces. So you know already how the class goes, how everything goes. But quickly I'll go over some important things before I start my lecture, okay? So all of you must know how to use the Blackboard because the important learning materials are posted on Blackboard. So you know this is the college website. So if you go to the top upper right corner, you see my STLCC, and if you click on that, you will open this buttons, and here you see the Blackboard, which is the most important thing. So you will log in Blackboard using your ID, which is the first part of your email and password that you have to create, okay? So using your ID and password, you will log in Blackboard. That will take you to this page where you will see the list of the classes you have signed up for this semester. Here you see the list of the classes I am going to teach in this semester. And your section is Bio 208-302. Okay, so if you click on that, it will take you to this page where you see in the left panel a lot of things. The most important things are course content and the lab materials here. Okay, so under course content you will find the syllabus, lecture and lab schedules. All those materials are already posted, so you see there. It is a good idea to print out this information and put in your folder or binder. So you will know when the exams are, which lab will be on which day. Those information will be always with you. So that's a good idea. If you want, you can do that. I like that. So quickly, I'll go over a couple of things here in the syllabus. This is my name. Almost all of you know. Class hours. You have 2 lectures. And 1 lab in each week. So the contact information is here. You can call me. This is my office phone number. Email me, email address, and this is my office room number. So you know, only one building we have here, so it's very easy to find me. Room number 228, okay? If I am in my office. We attend in meetings and other things. So you are always welcome to contact me. You can call me, email me, or directly you can come to my office if you want to talk to me. Your textbook is Human Anatomy and Physiology by Mariam, the same one you have used in NP1. If you are from another campus, then you might have to buy this one. And lab manual from this actually summer, I have created the lab manual, so I'll post the lab materials online. You don't have to buy any lab manual, but if you have one from NP1, you can actually get some help from that one, or you can sell it. It's up to you. Okay? But if you don't have, don't buy it. You will get the materials. I'll post. Still, it's on the process, in the process of creating, but you'll get it online. Okay. Read this section. It says which materials will be covered in NP2. Then the exams. There are 5 lecture exams and 3 lab exams. Each exam has 100 points, so total 500 points from lecture exams, 300 points from the lab exams. Okay, and then I'll average your score, and if your average percent is 90 to 100, your grade, letter grade will be an A. If it is between 80 to 89, your grade will be B, and 70 to 79 will be C. I hope you won't go below that. Okay, I will stay within top 2 or 3. If you need any special accommodation, you can talk to me or in the office downstairs. Inside the classroom, we follow some rules. Everywhere you follow some rules. When you drive, you follow some rules, right? Inside the classroom, the main thing that you have to keep in your mind is don't disturb others. Behave in a way that does not bother others. Make sense? So that is the main thing here. So during my lecture, if suddenly your phone rings, accidentally that can happen, right? It happens to me too, but you should be careful. Okay, you turn it off, keep it in silent mode. If you think you will get important call, you might get important call, keep it in silent mode. When you want to talk, you can go outside. Okay, I won't mind if you leave the class during my lecture, but don't leave in a way like you are, celebrating it or making big noise. Don't do that way. Just quietly go and come back. So that's fine. Okay. So those are the few things I wanted to share. Then you see here, lecture and lab schedules. Here you see 5 lecture exams, and you know the dates already except the final one. That one we'll announce later. Then we'll try to follow this schedule as listed, but sometime we'll go slow, sometime fast, but we'll try. Lab schedule will follow exactly as it is here. So there are 3 lab exams, and you already know the dates. The final exam will be on December 10th. That's the lab exam. And the lecture exam will be the following week. Okay. If you have any question regarding the syllabus, you can ask me now. If not, then I will go to my lecture. Okay? Another thing, always feel freedom to speak, ask questions, talk to me. Okay, don't feel shy or don't be afraid of asking questions. Okay, it is very important that you clearly understand the things. Okay, So today's topic is the blood. That's the first chapter we'll cover. So in this chapter, today first we'll talk how to define the blood. What's the definition of the blood? Then we'll talk about the general properties or characteristics of blood. Then we'll talk about the functions, important functions of the blood. Okay? Then the composition of the blood. That means blood components. What are the components present in the blood? Then we'll talk about the process of separation of different blood components. The way we separate different blood components or the method is called centrifugation. So we take the blood in a test tube or glass tube and centrifuge, spin it for a few minutes. That separates the formed elements and the plasma. Formed elements are the blood cells. Three types of blood cells are present in the blood: red blood cells, white blood cells, and platelets. Okay, so those are the formed elements, the blood cells. And plasma is the liquid part. So centrifugation will separate the formed elements and the plasma. So those are two main components of the blood. Plasma is the liquid part and formed elements are the cells. Three types of cells. Now, first we'll talk about the plasma part of the blood. Plasma is the liquid part and plasma is mainly water. 92% volume of plasma is water. So 92% of plasma is water. And also plasma has proteins. And proteins in the plasma are called plasma proteins. Make sense? Proteins in the plasma are plasma proteins. We will see what are the important plasma proteins present in the plasma. Okay? We will talk about the plasma proteins, their origin and functions. Then we will talk about the formed elements. That means the blood cells, red blood cells, white blood cells, platelets. Okay? First we will talk about the red blood cells. Morphology, that means external features. Morphology means external appearance. If you see a red blood cell, what's the shape, right? What's the color? Those are the external features. Then we'll talk about the maturation of red blood cells. After the red blood cells are produced, how they get matured. Then we'll talk about the destruction of the red blood cells. Red blood cells stay alive for 3 to 4 months. 3 to 4 months. That means 90 to 120 days, right? Then after that lifespan, the red blood cells are destroyed. So we'll see what happens after the destruction of the red blood cells. What's the fate of the red blood cell? So those are the things we'll talk today. First, definition. Blood is a special type of connective tissue that circulates inside a closed system. Blood circulates inside a closed system. So three things here. Number one, what type of tissue? Blood is a connective tissue. Why blood is a connective tissue? Because it comes from mesenchyme. All connective tissues originate from a special tissue that is called mesenchyme, also called mesenchymal tissue. You know that bones are connective tissue, cartilages are connective tissue, fats are connective tissue, blood is also a connective tissue. Now, why all those different structures are connective tissue? Because all of them come from the same source, single source. Now, why we say blood is a special type of connective tissue? Because it is the only connective tissue which is in liquid form, circulates. Other connective tissues are in solid form. Okay? And closed system is the heart and the blood vessels. Heart and the blood vessels together form the closed system inside which the blood runs, circulates. General characteristics or properties. Color. Arterial blood is bright red. Venous blood is dark red. Why is that? Where you have more oxygen, arterial or venous blood? Where you have more oxygen, in the artery or in the vein? The artery, right? So arterial blood is bright red because of more oxygen. When oxygen binds with hemoglobin, the hemoglobin gives bright red color. Make sense? Okay. When the oxygen leaves the hemoglobin, carbon dioxide binds, then the hemoglobin turns dark red color. Make sense? So that's why arterial blood is bright red, but venous blood, where you have more carbon dioxide, less oxygen, is dark red. pH of blood is 7.35 to 7.45. That means slightly alkaline. Make sense? What is the neutral pH? 7.0, right? So if it is above 7.0, it is alkaline. Below 7.0 is what? Acidic. So your blood is what? Acidic or slightly alkaline? Slightly alkaline, right? Because it is slightly above than 7. Now, it is good that your blood is not acidic. Make sense? If your blood is acidic, then the blood will kill all the cells, tissues, right, in your body. So it's good it is not in acidic side. Viscosity. Viscosity indicates the thickness and stickiness of the blood. So just know that viscosity is the thickness and stickiness. So blood is thick and sticky. That means the viscosity is high, right? Blood is thick and sticky. Temperature, slightly warmer than the body. So your blood is warmer than the body. The temperature of blood is 38 degrees Celsius. Volume of blood. In adult male, the total volume of blood is 5 to 6 liters. Total volume. In adult female, the blood volume is 4 to 5 liters. That means in female body, the volume of blood is what? Less. Why is that? Smaller. That is number one. The size, average size is smaller, right? Number two, even if you take same sized male and female, in male body the blood volume is more because of the amount of skeletal muscle. In male, the muscle is more. In female, fat is more than the male. Remember, muscle has more blood flow than the fat tissue. Okay, so that's why the volume is higher. Functions of blood. Number one is transportation. Blood transports the gases. Which gases? Oxygen and carbon dioxide, right? Blood transports oxygen and carbon dioxide. Number two, blood transports hormones. Takes the hormones from the gland to the target, site of action. Blood transports metabolic wastes, harmful chemicals. You know, metabolism occurs 24 hours, right? Continuously in your body, metabolism is occurring and harmful chemicals are being produced. They must be taken to the kidneys and lung. Blood takes the harmful toxic chemicals to the kidneys and kidneys excrete them through the urine. Which harmful chemicals are taken to the kidneys? Urea, uric acid, ammonia. You can write down those harmful chemicals. Those are taken from the tissue to the kidneys by the blood, and then kidneys will filter and excrete them out. Urea, uric acid, ammonia, those are really harmful and are being produced continuously. Blood takes nutrients to the tissue. So after you eat the food, nutrients are absorbed and taken to the tissues. So those are the things transported by the blood. Gases, right? Hormones, metabolic waste, nutrients, right? Anything else I said? Yeah. So those are the things. Now, regulation of pH. Blood also maintains the pH of the body. How? Blood has buffers. You know, buffers regulate the pH, right? Buffers regulate the pH. Blood has certain buffers called the blood buffers, and those buffers maintain the pH. So your body will not become too acidic or alkaline. What are the blood buffers? Important blood buffers are bicarbonate buffer, phosphate buffer, and another buffer that is called protein buffers. So these are important blood buffers, and these buffers keep the pH constant, unchanged. Coagulation, another important function of the blood clot formation. Coagulation is the process of blood clot formation of blood clot. And you know that by forming blood clot, blood stops bleeding, right? So bleeding is stopped by the formation of blood clot. So loss of blood or fluid. Defense, immunity is another important function. Just know that two components of blood are very important to provide immunity. One is the white blood cells. They fight against the microorganisms or antigens. Which cells? White blood cells, right? And another component of blood is also important, that is the antibody. That plasma protein is called globin, Globin, globin. Okay, so those are two components of blood that provides immunity. And globin is circulating antibody. You see there, circulating antibody, that's the globin. Stabilization of body temperature. How? If your body temperature increases, then what will happen? Vasodilation will occur in the blood vessels of your skin, everywhere. In the skin, the blood vessels will dilate. Make sense? Get wider. So more or less blood will flow in the skin. If blood vessels dilate, gets bigger, more blood will flow, right? So in the skin, the blood vessels will dilate, vasodilation will occur, so more blood will flow on the body surface. Make sense? In your body surface. So heat will easily get out from the blood. That's the main way how the blood brings the body temperature back to normal. Helps in the excretion of harmful chemicals in our body, from our body. As I have already mentioned, urea, uric acid, ammonia, carbon dioxide, those are the harmful chemicals. Those are taken to the kidneys or lungs and are removed from the body. Do you have any question now? Okay. Composition of the blood. If you separate the components of blood by putting the blood in a test tube and centrifuge it for a few minutes, what will happen if you take 100 milliliter of blood and then centrifuge for a few minutes, what will happen? The formed elements, that means the blood cells, will settle, will go to the bottom, settle down in the bottom, and the liquid part will go to the top. So this is how we separate the formed elements, that is the blood cells, and the plasma, liquid part. Okay. In normal blood, the formed elements, the volume of formed elements is 45%. About 45% volume is formed elements, and plasma forms 55%. Okay, so in 100 milliliter of blood, about 45% volume is the formed elements, and 55% is the plasma. Make sense? Now, formed elements consists of red blood cells, RBC, white blood cells, WBC, and platelets. So those are the blood cells, right? If you see the number of red blood cells, count the number of red blood cells. Red blood cells in 1 microliter, red blood cells are in millions. White blood cells are in thousands. Okay, so which is more? Red blood cells. 100 times more. So now if I ask you, this part is mainly given by which cells? Red blood cells or white blood cells or platelets? Red blood cells, right? Because red blood cells are millions in number. White blood cells and platelets are thousands. Make sense? So this part is mainly given by the red blood cells. Is it clear? Okay. So when you will centrifuge, what you will see that a thin white coat is formed here. That is called Buffy coat. That a thin layer, white layer, is formed here, BUFFY, and this is actually formed by white blood cells and platelets. Only this part. And this part, whole part here, is formed by red blood cells. Because we know that the number of red blood cell is much higher than the white blood cells and platelets. Okay? And this volume of formed elements is called hematocrit. Hematocrit. So hematocrit is the volume of formed elements in the whole blood. So hematocrit is the percent volume of formed elements in the whole blood. Normally 45%. Okay. Hematocrit is also called, another name is packed cell volume, PCV, packed cell volume. Makes sense, right? Because all cells are packed here in the bottom part. That's the packed cell volume, or PVC, PCV, or hematocrit. Same thing. Okay. Now, plasma is the fluid part or liquid part, and mainly it is formed by water. 92% volume of plasma is water. 7% is plasma proteins, and 1% is other solids like antibodies, hormones. Those are in the metabolic waste. Those are present in the plasma. So plasma proteins form 7% volume of plasma. We'll see what are the important plasma proteins and their functions. So here you see this is a nice chart. It is showing different components of blood. First you see after you take blood in a test tube, if you centrifuge, you can separate the plasma, which will go to the top, and the formed elements in the bottom. Plasma is about 55%, formed elements is about 45%. Then you see the composition of plasma. 92% is water, 7% plasma proteins, 1% other solids. Now, important plasma proteins. 3 plasma proteins are very important. What are those three plasma proteins? Albumins, see the top box, albumins, globulins, and fibrinogens. So those are three important plasma proteins. Remember those names: albumins, globulins, and fibrinogens or fibrinogens. I'll just mention one or two functions of each. Albumins are responsible for the colloid osmotic pressure. Oh, what is this? Something sticky. Put it here. So, albumins are responsible for colloid osmotic pressure. Osmotic pressure. What is this? This pressure helps to keep the blood in the center of the blood vessel. So this is the blood vessel, and you know that blood passes through the blood vessel, and when the blood passes through the blood vessel, this pressure tries to keep the blood in the center of the blood vessel, so the flow will be fast. If the blood components try to go outwards towards the wall, then the blood will get slow. Make sense? So colloidal osmotic pressure will keep everything together in the center, so the blood flow will be faster. So that's the function of the albumins. Globulins. Are responsible for immunity. The globulins, which is mainly responsible for immunity, is called immunoglobulins. Okay, so immunoglobulins are the globulins that help in the immunity or defensive functions. So globulins are mainly responsible for immunity, just write it down. Globulins also help in transportation of chemicals like hormones and metabolic waste. Those are also transported by the globulins. So two functions, immunity and transportation. Fibrinogens are responsible for blood clot formation. So fibrinogens, blood clot formation is helped by fibrinogen. So those are important functions of those three types of plasma proteins. Just remember those three, should be fine. Origin of plasma proteins. More than 90% plasma proteins come from the liver. So liver is the main source of the plasma proteins. Small amount of plasma protein comes from a cell that is called plasma cell. So plasma cells produce a small amount of plasma proteins. Which type? Globulins. So just write it down that globulins, some globulins come from plasma cells. And those are the two main sources of plasma proteins, liver and plasma cells. Formed elements are blood cells. Three types of blood cells we have in the blood. Red blood cells or RBC, also called erythrocytes. The word erythro means red. and site means cell, you know that. So red cells, erythrocytes. General properties of red blood cells. The color of red blood cell is what? Red. That's why it is called red blood cell. And why the color is red? Because inside the red blood cells you have the pigments called hemoglobin, which is red colored pigment. So that's why red blood cells are red, because of the presence of red pigments, hemoglobin. Make sense? Shape. Red blood cells are round but flat. Round but what? Flat, not spherical, flat. Why the red blood cells are flat? Because no nucleus is present in mature red blood cell. Enucleated, absence of nucleus. Make sense? Since there is no nucleus, the cell can get flat. If there is a nucleus in the center, then it cannot get flat. Make sense? So, red blood cells are enucleated or non-nucleated, and they are flat, round. And outer part of the red blood cell is thick, and inner part is thin, like a membrane. Thin membrane is in the center part, but outer part is thicker, and that's the rib, outer rib. So, in a mature red blood cell, you don't have any nucleus, but you have hemoglobin. That is important, right? Hemoglobin is present. Hemoglobin binds with oxygen and transports the oxygen. So that's the main function of red blood cell, transportation of gas. Now, having the flattened shape of red blood cells have some advantages. What are the advantages of having flattened shape? Number one, they can easily get folded, get smaller, and pass through the narrow capillaries. You just think that you have a soccer ball. If you fill the soccer ball with air, you won't be able to fold it, right? But if you take all the air out, make it flat, then you can easily make it smaller, right? So red blood cells can get folded, squeezed through the narrow capillaries. Even they can pass through the capillaries that have smaller diameter than the size of the red blood cells. They can pass through that. Number two, having the flattened shape, the hemoglobin can easily bind with the oxygen and release the oxygen. How? Just think that if it was like a round sphere and the hemoglobin is here, oxygen has to pass through the plasma long way to get attached to the hemoglobin. Hemoglobin is here. But actually is what? Red blood cell is flat. That means it's like this. So hemoglobin is here. Now you see oxygen will be able to get attached and detached easily. Make sense? Because hemoglobin is attached to the cell membrane. So from outside, oxygen will quickly get attached to the hemoglobin and can quickly get out, in and out. But if it was like this and hemoglobin is here, then oxygen has to travel through the plasma a lot, right? So hemoglobin comes very close to the cell membrane because of the flattened shape, and oxygen can easily get in and out. So that is another advantage of of having flattened shape. Here you see the shape and structure of a red blood cell. See the most right picture. Outer rim is thick and center part is thin. You don't need to remember the numbers, just know that the shape is like a donut. Usually donuts have hole inside, right? But in this case there is no hole but a thin membrane in the center. One important property of red blood cells is rouleau formation of rouleau. What is that? Since the red blood cells are flat and round, they can easily get attached to each other, stuck together and form a column. See here, like form a column like this. They can easily get attached to each other. It's like, you know, when you go to a big dinner party, you see the plates, dinner plates are stuck together, right? Piled together. So red blood cells can easily get attached to each other like this, and that is called the rouleau. Count of the red blood cells. In 1 microliter of blood, 1 microliter is a lot of blood or very small amount? A lot or a small? Micro means 1 milliliter is 1,000 of a liter, right? And 1 microliter is further 1,000. It's a very small amount of blood. Has millions of red blood cells. Remember, I told you. So, in 1 microliter of blood, in adult male blood, you have 4.5 to 6.3 million red blood cells. Can't imagine. In female, it is less. The count of red blood cell is 4.2 to 5.5 million. Why the red blood cell count is more in male than the female? Say loud. Why? What do you think? Why red blood cell count is higher in male than the female?
More muscle mass.
That is more muscle, that means the volume is more. That doesn't, if you take same amount, like once you take it out, 1 microliter from male and female, size doesn't matter, right? Because you are comparing same volume of blood. Why the red blood cell count is more? Number 1, in male body, metabolism occurs more metabolism. Metabolism requires more what? Oxygen, right? And red blood cells transport oxygen. So remember, where metabolism is more, red blood cell count